Hello everybody and welcome back. So, we are working on module 4 where we are discussing DNA damages and their mutations. In the last lecture we have seen how the UV induced DNA lesions are formed, how the photochemistry works, and how there are the nucleobases are undergoing structural variations or structural orientations that are responsible for inducing the mutations in the genomic DNA. So, we have seen that if you irradiate the nucleic acids, then primarily I have talked about the nucleobases that undergo different kinds of lesion formations. So, I will just quickly recap and then we will move on. So, if you have a dinucleotide with 2 thymine and thymine with a phosphate here, and if you irradiate against the UV light, primarily the UVC and UVB radiation, then the products that are generated are majorly the TPT, right, double bond T, this is CPD lesion, cyclobutane pyrimidine dimer, and mainly in the form of cis-syn, that is your major content, along with TTCPD with trans-syn geometry is minor. The other one is also minor, the cis anti, and then you have 6,4 TT or TPT lesion, that is also a minor product, and then this one was converting into the Dewar valence isomer, Dewar lesion, that is also the minor product. Similarly, if you start with CPC, both cytosins, the neighboring nucleobases are cytosins, UV radiation, then what you form is you first get the CPD lesion, which is deaminated into the uracil UUCPD and cis-syn, that was your major component, along with 6,4, one uracil and one should be cytosine UC lesion and the Dewar one, Dewar UC lesion. Similarly, TC can give rise to a variation of products, and all these compounds have enormous differences in their chemical structures. And we have seen how the minor compounds were highly mutagenic in nature. So, even if they are formed in very little quantity within the cell, they are highly mutagenic in nature, because their structural orientation is drastically different compared to the original nucleobases. So, they are kind of like poisons, less quantity or highly effective, very dangerous. So, a lot of research has been carried out to understand how these lesions are formed. Lot of methodologies have been developed to identify all these kinds of lesions. So, I have restricted myself only to the two kinds of lesions, oxidative lesions and the UV induced lesions. There are other kinds also. So, lot of studies have been performed to identify so various structures or various kinds of lesions that are produced, and their structures were elucidated. Sophisticated techniques, instrumentations like laser spectroscopy, high end spectroscopic techniques were used. LCMS, that is chromatography, liquid chromatography coupled to the mass spectrometry. LCMS, that is that studies the fragmentation patterns of the compounds, organic compounds. So, from all those studies, different kinds of lesions were isolated and their structures were elucidated. From those structures, we have tried to gain insight informations that what could be their properties, what kind of mutations they could induce. And in the last lecture, I had shown you a table where a total gene or total cell have been studied to know what kind of lesions or how many quantity of the mutations that have been actually formed due to the lesion formations. If you formed a lesion inside a DNA, if you damaged a particular nucleobase in a genomic DNA, how to know what kind of mutation that lesion is going to induce, how to know it experimentally. For example, I take a single stranded DNA here 5 prime 3 prime, and here there is a lesion. I am writing L for lesion. This can be any kind of lesion. Now, this lesion has been formed. Now, how to know what kind of mutation this lesion is going to induce or going to induct into the genome. So, a way to know is this, and here comes the organic chemistry again. Chemistry can play a major role in identifying or in knowing or in studying the formation of the mutations. So, what you can do is you can synthesize a whole length of the DNA in which you can chemically incorporate the pure lesion. So, you can synthesize a lesion in the laboratory in pure form and then convert it into a DNA monomer and then construct a DNA, this. You can construct this DNA incorporating a lesion at one point of space here or maybe again here. You can have one, you can have multiple of them. So, you can synthesize a DNA in which you know that your pure lesion has been incorporated into it, and you also know its location. So, next what are you going to do is, obviously you cannot put this gene into the living cell and study the replication, because in that case there is a very good chance that the lesion will be immediately repaired and you cannot find a trace of it. So, living cell study may not be very helpful here. What you can do, and that is what people actually do, is study this in vitro. In vitro replication means the PCR. So, you have this DNA, you use this DNA with the lesion in it as a template and try to do the PCR with other ingredients, of course, polymerase and DNTPs and all. Then you can know what kind of the complementary strand this is going to synthesize. So, polymerase will pick up a suitable, this is the complementary strand which you are synthesizing or which you have synthesized using the PCR, and then followed by sequencing of this complementary strand, that will tell you what nucleobase has been taken up by the polymerase opposite to this lesion, opposite to this lesion. And if you do the sequencing, then you can know the identity of these two nucleobases, and from there you can have the accurate idea of what mutation this lesion would induce, because the mutation would be the complementary of this nucleobase. So, that is how people find out how individual lesions, what kind of mutations that will be bringing in into the genomic DNA. So, now let us move on to the other part of it, that is how to repair the damages, how the cells adapt methodologies, or how the cells adapt processes to repair various lesions that are formed. Repair of DNA lesions or damages. And I have talked about 3 different methods that the cell adapts to repair the damaged sites of the in the genome. One is the base excision repair, this is called BER. Second one is nucleotide excision repair, that is your NER, and the third kind is mismatch repair. Short form is MMR, mismatch repair. So, depending upon the kind of lesions, one of this procedure would be adapted by the cells. So, let us start with base excision repair. As the name suggests, this means excision of a nucleobase. So, if you have the DNA, DNA is continuing, here is your base, right. So, if this base is damaged, I will write this as lesion, nucleobase lesion. So, this is the damaged nucleobase that has been formed in the DNA. Base excision means that it chops off the nucleobase part out of the DNA. So, this is your, this bond is your glycosidic bond. So, the repair mechanism, once it finds out that there is a problem here in the nucleobase, then it clips off the glycosidic bond and then it removes the lesion base out. So, you have a OH now here, so your DNA, that is your DNA. Base is out and you are left with the hydroxyl group here. Now, this DNA which devoid of a nucleobase in the sugar is known as A basic site. A basic site. or A basic DNA or A basic sugar, meaning that it is devoid of the nucleobase. So, only the sugar moiety is present here. So, that is the process of base excision repair. Obviously, it is done with a set of enzymes that we will see now. So, basically if you have a DNA here 5 prime to 3 prime, this is your skeleton, phosphate is there, and let us say here is the OH 5 prime. So, this is your 5 prime end lesion, here O and then phosphate and it is going out. So, your DNA after the base excision repair will look like at the beginning OH and this will go on. So, single stranded DNA without the base, A basic lesion. So, this is actually also a kind of lesion that I have not talked about. I have talked about only the nucleobases. So, sugar lesions, A basic lesions are actually formed in large quantity in cells also. So, now how the process looks like. Five prime to 3 prime, all the nucleobases, and here there is a lesion, let us say nucleobases, complementary strand 3 prime, 5 prime, base pairs, here there is something, the original one that was there against this, the base pair. Now, let us say this is your lesion or damage. Base excision repair mainly occurs of the DNA lesions which are oxidative lesions. Mainly adapted for the repair of oxidative lesions such as 8-oxoguanine, 8-OG, 8-oxoadenine and so on. So, the first stage is removing the nucleobase out of the DNA, and this is done by the enzyme. How does it remove? By cleaving the glycosidic bond. So, it cleaves the glycosidic bond, right. So, therefore, the enzyme is called a glycosylase. Glycosylase is the enzyme that cleaves the nucleobase out or that cleaves the glycosidic bond, and you are left with sugar. So, this is your sugar and A basic. Five prime, 3 prime, 3 prime, 5 prime, this. Now, what you have to do, there should be something incorporated, the correct nucleobase has to be incorporated in this position, then only it will be repaired. So, what the repair mechanism does, it does not take any chance, it does not take any risk. What it does, it will chop once this abasic site is formed, it will chop off a portion of the single stranded DNA containing the abasic lesion. So, this will remove or excise short segment of the single stranded DNA containing the abasic site in and around, little bit left, little bit right. So, this is just like surgery. If we have a wound, when doctors go for a surgery, they always remove a little bit in and around the wound area, so that the infection does not spread. So, same thing happens here. The repair system has to be very precise. So, after cleaving this thing up, what you will have is this 5 prime 3 prime and your whole DNA. This. Okay. This enzyme I can write here. This enzyme, which actually clips the DNA section out, is known as endonuclease. There are different types of endonucleases. So, one kind of endonuclease would be used to remove the section of the single stranded DNA that contains the abasic site. Now, when we are talking about the oxidative lesions, most of the times or a major portion of the oxidative lesions are the purine, are coming from the purine nucleobases, guanine and adenine. So, this site, the abasic site that we have been calling, is also known as apurinic site. Apurine or apurinic base, because it is now devoid of the purine nucleobase, and this endonuclease is known as apurine endonuclease. If the lesion is coming from the purine nucleobases, then this endonuclease would be used. Now, you have a empty space here, which you need to reconstruct, and certainly you have to synthesize this portion using the complementary strand as the template, and obviously here comes your polymerase. So, DNA polymerase 1 is used here, that will fill up this gap taking the 3 prime to 5 prime end or the strand as the template. So, now it will incorporate the correct nucleobases here. Now, there is still the gap here because it was synthesized discretely, and now you need to stitch them together. As usual, you need a DNA ligase. And now you have a completely repaired strand. With the correct nucleobase in place of where there was the damage originally, where the lesion was there. So, this is one of the processes, one of the three kind of process that I have talked about. Base excision repair. Excision means cleaving it off or removing, and this is mostly applicable for the lesions which are oxidative in nature. Key enzyme is glycosylase. Is the enzyme that actually removes the lesion, oxidative lesion. It removes the oxidative lesion. One kind of glycosylase that we have, the human glycosylase, is known as, that has a name HOGG1. There are many glycosylases. So, this is human 8-oxo, O is for oxo, and that inevitably means 8-oxo for guanine, 8-oxo guanine glycosylase 1, HOGG1 is the enzyme that actually repairs all the oxidative lesions, most of the oxidative lesions that are formed inside our cells, eukaryotic cells, and this particularly is the human. Okay, so that is about the rough sketch or the rough scheme of the base excision repair. Now, let us move on to the nucleotide excision repair, NER. In this case, base is not removed, but the whole nucleotide is removed. Let us start with a particular example. Maybe we can use the TT here, T and T double bond. So, here would be A, here would be A originally. Nucleotide excision repair is adapted for the lesions which are UV induced lesions. There are other kinds also. In our domain, whatever we have covered is the UV induced lesions are repaired through this procedure or through this process. UV induced DNA damage are repaired through NER. For example, TT, CC or CC becomes UU, uracil dimer, and so on, CPD lesions for example. So, what it does is, as the name says, that it cleaves off the nucleotide. Nucleotide means the base, the sugar, the phosphate, whole chunk of it. Because what happens, the CPD kind of lesions are not very well recognized. They look like, because you have seen the structures, there is only a cyclobutane pyrimidine ring in between them. Other than that, their whole other framework is almost the same as the original nucleobases. So, sometimes the repair system cannot understand whether this is correct or this is wrong. So, it does not take any risk, it simply cuts off a portion. First it chops off the part of the where there is the lesion part, that whole nucleotide part is cut off. 3 prime, I am keeping A here. By the enzyme, of course, is endonuclease. And then again, just like the other surgery we have seen, it does not take a risk, it removes a certain part of the sequence just to make sure nothing else has been infected. Five prime. So, you have a gap here that is created, 5 prime, 3 prime. Here was AA, this followed by the action of DNA polymerase 1 and number 2 is DNA ligase. That will make your original strand back. That is how this is your repaired strand. Repaired DNA. So, that is how the nucleotide excision repair works. Some of the examples of the nucleotide excision repair enzymes are, I will give you some names: UVRA. You do not have to remember the names. B, UVR, UV repair, basically. See, these are some of the enzymes that are involved in the nucleotide excision repair. The third category is mismatch repair. MMR. Mismatch repair is it may not be from a lesion. You have a double stranded DNA, but somewhere there is a mismatch of nucleobase, mismatch of base pair. So, if this is, I am writing this as triangle, this as a rectangle, so that they do not match each other, they are not complementary to each other. So, this is mismatch portion. Mismatch base pairs. Not necessarily they came from any particular kind of lesion, it simply has been occurred because of the mistakes during the replication process. Of course, mistake can happen anywhere. So, if there is such kind of mistake that against a thymine, a cytosine had been incorporated wrongly by the polymerase during the replication process, then what happens is, after a DNA is synthesized in the cell, after a replication process happens, then the cell has proofreading system, just like our own writings. Once you write a certain document, then you always do a proofreading, go through it again and again whether there is any mistake, spelling mistakes or anything other kinds of mistakes there. Similarly, cell also has that. So, once the replication is done, the proofreading system will scan through the entire gene and will try to find out if there is any mistake in its work. And here comes the mismatch. If there is any mismatch, if it is located by the proofreading system, then it requires to be repaired, and this is what the system is, mismatch repair. It is little bit different than the base excision and the nucleotide excision. One thing what I did not tell you is that our DNA undergoes methylation inside the cell, and that is a very natural process actually. DNA methylation is a kind of natural process that happens to DNA. Normal our DNA as well as microorganism DNA of the prokaryotes as well. This is sometimes to protect the DNA so that they cannot be recognized by the enemies. So, the methylated sites are the target here, are the recognition sites in this case in the mismatch repair. So, let us say there is a DNA methylation on this base here, and there is another over here. So, in this mismatch repair, what the repair system does or what the enzyme does, it will recognize the two closest methylated points closest to the mismatch, and then it will cleave off a certain part closer to these methylated sites, and it will give you here. So, it will recognize this and it will chop off in this part 5 prime. Here is your methylated site, methylated. So, here it will cleave off. On the other hand, you have another methylation here. So, it will cleave from in this site 3 prime, and you get empty space. It will make one cut here, it will make another cut here, and the whole chunk is out. So, you have something like this void that is created, and then it will be filled up. Of course, now the enzyme, because there are two cuts here, the enzyme is exonuclease for this cut actually here. This enzyme is exonuclease. So, you need SSB protein. Of course, that you had required the SSB proteins and all sorts in the previous ones also, helicase to stabilize, and then followed by polymerase, and then it will be synthesized discretely. It was cut in the methylated site. Here is CH3 and here is your CH3, and followed by DNA ligase will ligate it. CH3 and CH3. You will get your mismatch repaired. So, that is what is the mismatch repair system. So, these are the prime processes or primarily these are the three methods that are widely occurs inside the cellular systems to rectify the lesions or to rectify a mismatch. Now, there is another kind of repair, and that is done by a certain enzyme called photolyase. So, this happens in the plant's system. All plants or most of the plants have this enzyme called photolyase. Most of the plants have, lower organisms also have. Lower organisms also have this enzyme photolyase. Of course, you can understand the leaves in a plant, they undergo or they experience or they are exposed to radiation much more, and also they harvest light for their photosynthesis. So, there also the photochemistry occurs in the DNA that forms the CPD dimers, CPD lesion 6,4 photoproducts and so on, also happens to the lower organisms as well. And since the plant can harvest the light, it can use the radiation for the repair also. So, the photolyase enzymes, there are many classes of photolyses. CPD photolyase is there, that is cyclobutane pyrimidine dimer photolyase. There is 6,4 photoproduct photolyase that had been isolated. So, we have done some work on that field also. So, photolyase is the enzyme or one of the few enzymes actually that requires light for its action. So, there are few enzymes that uses light as a reagent for its function, like our eyes have cryptochrome. Cryptochrome is a photosensitive enzyme. The photolyase is one such kind. So, this is one of the structures of photolyase, which shows that it binds to certain coenzymes, it requires certain coenzymes. This is MTHF, methyl tetrahydrofolate. Here is the FAD, flavin adenine dinucleotide. So, it requires certain coenzymes or cofactors for its function. What photolyase does is actually you have formed the CPD dimer, cyclobutane pyrimidine dimer, through a 2+2 cycloaddition reaction. What the enzyme does is does the reverse of it, cycloreversion. So, 2 pi plus 2 pi cycloreversion, that is the job of the photolyase. Since it is doing a reversible process, of course, the radicals are involved, because when the photochemistry happened, the photochemistry occurs through homolytic cleavage, which means through the radical formations. So, which means after the CPD is formed, if you want to break the CPD, you need to inject electrons here to create radicals, and that will do the subsequent chemical reactions. So, you need to inject an electron there, and that electron comes from the coenzyme flavin adenine dinucleotide, FAD, and the electron to the FAD actually comes from the methyl tetrahydrofolate, the second coenzyme. I am just showing you the mechanism of action, how it works. So, photolyase binds with this as well as this, and it harvests radiation from sunlight. Photolyase absorbs the radiation from sunlight, and this radiation is not the UV, this is the normal radiation, visible light. Chlorophyll also absorbs the visible light during photosynthesis. So, photolyase harvests or photolyase absorbs the visible light through this MTHF, because MTHF absorbs in that range of the spectrum, and then MTHF, this is MTHF with radiation, this is visible, it goes to the excited state MTHF star, and it forms this and with a negative charge here. That funnels the energy to FAD, because if FAD has to transfer an electron, it needs to be reduced to FADH minus. One negative charge has to be created here, then only it can inject the electron, and it has to go to the excited state. That is why you need the light. Radiation makes the electron jumps to the excited state. So, visible light is absorbed by MTHF and then funnels to the FADH. FADH goes to the excited state and then gets reduced to minus, FADH minus. Why not directly light is absorbed by FAD? Because FAD usually absorbs in the UV range, but you do not require UV range light, because the plant is constantly harvesting the natural visible light. So, it channels the energy here, and now FAD transfers one electron to the dimer. Therefore, you get this. If you inject one electron, this is a keto group, obviously it will be homolytically open, it will be one dot here, and then the electron, extra electron is there. Extra electron of course will go to oxygen because it is more electronegative. So, we will get O minus here and a radical here. Now, once this radical is formed, it can trigger the opening of it here to here. So, unwinding, basically reversible process. It forms this, ultimately it generates this radical, and that radical again goes back, the electron again goes back to the MTHF, and you have your repaired DNA, or the CPD gets repaired. So, that is how the photolyze acts for the CPD lesions. So, here comes how the study was. How did you know all these processes is happening, that MTH is transferring to FAD, FADH is now doing the job, and all sorts of things. These were studied in through spectroscopic techniques as well as through EPR, because EPR can detect single electrons. So, here you do not have to remember this, this is just for your information, that if you have initially you have the active site of the protein contains a tryptophan and a tyrosine next to FADH. So, FADH goes to excited state, then tryptophan, tyrosine is there. Look at the time scale, it is only 500 microsecond it takes to reduce the FAD into the FADH minus, then tryptophan, then tyrosine. Then the radical is transferred to the tyrosine here from FADH minus in 50 microsecond. That is one kind of mechanism, and from tyrosine this will be transferred to the lesion, CPD lesion. That is what the function of the enzyme itself, apart from the cofactors. A second mechanism is FADH in another kind of photolyze. So, there are two kinds of photolyzers for CPD itself, photolyze 1 and photolyze 2. So, other photolyze has tryptophan, 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 3 tryptophan moiety in the active site of the protein, active site of the photolyze, and that is close to the FAD. If I can go back, here is the FAD and here is the active site of the protein, W here, this, the amino acids. So, FAD goes to the excited state through the light, goes to radical formation, then it gets reduced through the tryptophan, then tryptophan injects, pumps the electron to here, it gets there, again pumps the electron to here, it is kind of like electron hopping or tunneling of electrons, you get this, and you get the radical on the tryptophan finally. So, studies have also been done using only tryptophan. If you can make a radical on tryptophan, it can also do the repair process. So, lot of molecules had been synthesized that covalently outside the enzyme, just without using the enzyme, FADH conjugated to tryptophan, conjugated to the tyrosine, and the repair also were studied, and through them also this kind of mechanism were developed. Alright, so this is other kind of the repair system through the photolyze. So, with this I think I will conclude module 4, that in this whole module we have talked about the genomic mutations. How are they formed? What are the chemical bases behind the formation of mutations in the genomic DNA, and how the chemical structures or alteration of chemical structures can govern the biological processes again. So, first is genomic mutations are the cause of uncontrolled cell division in tumor cells. Nearly all tumor cells have the mutations in the genome, and that is the primary cause of their uncontrolled cell divisions. For example, in our cell, when cell division happens, there are two genes that control the cell divisions. One is called oncogene. The role of oncogene is actually it accelerates cell division. So, it will favor the cell division. Favor cell division. Favor cell division means favor the divisions of or favor the replication itself. And there is a second gene known as tumor suppressor gene. Tumor suppressor gene that actually reduces the rate of cell division. Rate or the speed of cell division. So, one gene is accelerating cell division, the other gene is decelerating the cell divisions. Now, when these two genes act properly, then we have a controlled cell division that happens all the time in our body. Now, if there is mutation happens in either of these two genes, for example, if the tumor suppressor gene is mutated, then it cannot function again anymore. I had given one example that P53 gene. P53 gene is a kind of tumor suppressor gene. So, if the tumor suppressor gene is mutated, then it cannot function, it cannot stop the cell division, or it cannot control the speed of the cell division. In that case, what will happen? Oncogene will function only and your cell divisions will be more and more. That is where the uncontrolled cell division happens. That is what happens in the tumor cells. So, genomic mutations are the cause of uncontrolled cell division in tumor cells. Mutations occur due to damage of the genomic DNA when exposed to various internal as well as external agents such as the radicals. So, reactive oxygen species or ROS produced in cells, they create oxidative DNA lesions, and all these ROS chemicals are produced during metabolic process or also the other processes. Reactive oxygen species, for example, the peroxide radicals, even superoxide radicals, hydroxyl radicals, they are highly reactive in nature. So, they are responsible for oxidative cell divisions, oxidative DNA lesions. Oxidative DNA lesions are highly mutagenic in nature. You have seen how 8-oxoguanine, because of its structural property, it has totally changed its base pairing ability, and therefore, the capacity of inducing mutations is high. UV induced DNA lesions produces photoproducts. So, we have seen the photochemistry or photochemical lesions that are generated due to UV radiation of the DNA and are a major cause of skin burn and skin cancer. So, when you are exposed for a prolonged time through sunlight, if you especially go to a high altitude area, trekking for example, then you will most often see that you have to use a heavy amount of cream on your surface of the skin to prevent it from sunburn. So, sunburn basically is because of the formation of the UV induced DNA lesions, different kinds of DNA lesions, and skin cancers are most of the cases are because of the DNA damages that are formed due to UV light. Most often the DNA lesions are quickly repaired, as we have seen in cells, primarily by base excision repair, nucleotide excision repair, or the mismatch repair process. Along with, of course, the I have added one additional one, that is the photolyse, that is mostly for plants as well as the some of the lower organisms. They have this kind of repair systems which require visible light to do the reversible cycloreversion process. So, yeah, that is all for this module, that how the mutations happen and what are the cause of the mutations and what is the effect of the mutation, that is the uncontrolled cell divisions. So, with this I will conclude this module, and then next we will go to module 5. Thank you.